These webinars have replaced the regional forums during the pandemic. We're hoping to be back to normal regional forums next year. My name is Simon Newitt. I am an extension horticulturist based at Nambour with the Queensland Department of Agriculture and Fisheries. And these webinars are part of the Avocado Extension Project, AB 17005, called Avocado Industry Development and Extension, delivered jointly by Avocados Australia and the Department of Agriculture, and funded by Hort Innovation, using the Avocado Research and Development Levy, co-investment from DAF, and contributions from the Australian Government. Right. Firstly, please, some housekeeping. Please keep your microphones muted and your videos off. I know we've been, some people keep switching back on again, so you don't need your video on to see the screen, and we prefer, or we insist that you have your microphones muted too, otherwise it just interferes with the presentations. So thank you for that. Just a note too, if you've joined in without using your RSVP name, please attempt to change this so we can identify you. So for example, you might just have your iPad called John's iPad. Well, we don't know if there's a lot of Johns around, so if you could, it needs to be changed to John Smith. Now, the way to do that is if you go to your screen and find where it says participants, click on there, find your name and hover over there, and you'll see a little option comes up with more, a more button. If you click on that, you can add your real name. I appreciate that. Thank you very much. If you can't do this, on your chat box, we might send you an individual message. Okay, so with the microphones all off, if you do have a question, we ask you to type it into the chat box. There's a little icon there showing on the screen. Type your question there, and to make sure it's sent, you then just hit return on your keyboard. And finally, expect a slight delay as we change from one presenter to the other, because we're all in different parts of the country. Right, just a little run through the agenda. So after my brief introduction, we're going to welcome Dr. Neil Wilson from Metagen, and he's going to talk about, his topic is exploring soil microbiology in avocado production. So he'll talk for about 35 minutes. And then we've allowed 10 minutes for questions. As I say, use your chat box. If we don't get through all the questions, they will be recorded, and we'll ask Neil to answer them via email, and they'll be noted in the minutes. And then we'll hand over to the team from the University of New England, headed by Professor Andrew Robson, with support from Shephard and Joel McKenzie. And they're going to talk about improved orchard mapping and yield forecasting. So John Tice won't talk to us today because he gave an update at the last webinar and assures me that nothing much has changed. So thanks, John, for allowing your space to be used by some other speakers. And then just finally at the end, we'll have a conclusion and feedback. So just like to introduce our first speaker, Dr. Neil Wilson. He's the Director of Research at Metagen. So welcome, Neil, and I'll hand over to you. If you'd like to share your screen, Neil. Over to you, Neil.
Okay, can everybody hear me?
You've only just come on, Neil, so if you could start from the beginning again.
Sure. Okay. Can everyone hear me?
Yes.
Okay. So I guess the topic of my talk really is actually, the topic I like is the importance of the little things in life. This is a talk primarily about microbiology. So I'll start with a bit of a presentation summary of what I might talk about. And the first thing is, I guess it's my standard spiel to anyone who will listen as an enthusiastic microbiologist, that microorganisms are truly amazing creatures and really should be appreciated by everyone. Then I'll talk a little bit about this idea of the microbiome concept. This is becoming a very popular idea about the power that microbes have to influence the health of plants and animals and many other environments. Then I'll talk a little bit about the role of the microbiome in soil and plant health, followed by some direct examples straight from the scientific literature of the sorts of things that that the microbiome can achieve for soil and for plant health. Then I'll give a bit of a snapshot of some recent research we've done at Metagen, followed by approaches for actually quantifying soil health. So if we want to measure soil health, if we want to get an idea of, if we want to be able to quantify it, how do we actually go about that? And then finish off with some of my own thoughts about technologies of the near future in agriculture. So really, with the microbiome, it's not, too much of a reach to say that a revolution is upon us. So if I was to say to you, give me a plant, and without doing any kind of genetic engineering, I'll give you back a plant that is resistant to pathogens or resistant to heat or insects or salt stress, flood or drought. While that may be a bit of an overstatement, it's fair to say that the microbiome plays a role in protecting plants or giving the plants the ability to deal with all of these different things. So really, this is the power of the microbiome. And in my opinion, it's the future of biological farming. So there's a famous entomologist who made, relatively famous, the statement that it's the little things that run the world. And he was really talking about ants. But he said later in his life that if he could do it all over again, he'd be a microbial ecologist. And that was because he didn't have to go very far. He could just go into his own garden and find these fantastic creatures living there. And even though they're tiny, the sorts of things that they do mirror what you'd find in a real ecosystem. So they parasitize each other and they form symbioses and they camouflage themselves and predate on each other. So even though they're tiny, they still do all of the things that larger animals do. So really, it's the really little things that run the world. It's, microbes are by far, unquestionably, the dominant form of life on Earth. So in terms of abundance, there are far more microbes on Earth than there are stars in the entire universe, around 1 million times more. In terms of diversity, microbes are by far the most diverse creatures on Earth. So this picture in the middle of the slide there is what we'd call an evolutionary tree. It represents diversity, but also evolutionary time. So the little green box you can see there, that covers all of the diversity of all of the known plants, all of the known animals, and all known fungi. The rest of that tree are all microbes. So by far, in terms of diversity, the majority of life on Earth are microbes. Then what all of that means is that really in terms of function, given that you have that microbes are so abundant and so diverse, is that they are functionally the most diverse creatures as well. So there are a number of things that microbes can do that nothing else, that no other living creature can do. Nitrogen fixation is a really good and relevant example. There's all sorts of bioactive compounds that they can make that no other animals make. An example there is almost all of the antibiotics that we use are microbially derived. They're made by bacteria or made by fungi. They're also the most adaptable creatures, so they can live at extremes of pH and salt and heavy metals and temperature. They're also the fastest growing organisms on Earth, but also the slowest growing organisms on Earth. So they're diversity, their function, their abundance is really amazing. And that's what makes them probably, in my opinion, the most interesting living creatures on Earth. Now, the microbiome concept, some of you may have heard of this term microbiome. If there was one technical term I could get that I'd like everyone to take away with them, is this idea of the microbiome. All the term means really is a microbiome is just an ecosystem of microorganisms. And the term is now used to describe microbiomes of a whole heap of different environments. So you can have the ocean microbiome, the soil microbiome, human microbiome, or plant microbiome, and then a whole heap of others. You can name almost any environment and it has an associated microbiome. You can also divide up, so for example, the plant has a number of different microbiomes. There's a distinct microbiome of the leaves and a distinct microbiome of the roots, for example. The point here is really that every environment on Earth has an associated microbiome. The microbiomes are the foundation of plant and animal health. From a microbe's perspective, there's actually not a lot of differences between a human and a plant. You can view the root system of a plant as being quite similar to the digestive system of a human. It's an organ that's made for absorbing nutrients, for absorbing water. You can also view the skin of a person or the skin of an animal as being similar to the leaves of a plant. It's a protective surface that mostly is trying to shield the internal parts of the organism. There's a lot of similarities in terms of the structure of these different environments as well. So root systems and the gut are open systems that have a very high surface area. There's lots of niches. So there's lots of different kinds of environments, within and around and on the surface of plants and animals. And this is because there's lots of gradients in things like pH and oxygen availability. It's also noteworthy that plants and animals interact with the microbiome to shape it. So they're not passive in this. So plants send out signals that specifically recruit particular microbes. And the outcome of this is that the interaction between microbes and larger animals and plants has a whole range of effects, from protection of the host from disease or resistance to stress, but also the provision of nutrients and growth factors and things like that. It's interesting that even though you'd think of humans and plants, or animals and plants, as so different, it's the same 4 groups of bacteria that dominate these environments. So the take-home message here is that really microbial life underpins the health and well-being of all multicellular life. So the concept of soil health is not really that straightforward. It's actually, in the scientific literature, it's still, the definition of soil health is kind of difficult to pin down. But it's pretty much agreed that there are definitely physical components, chemical components, and biological components to that. And a healthy soil is a product of all of those factors working together. The biological factors are arguably the most important for soil health. And the biological factors, common ones that you could think of, is the diversity of those microbes that are living in the soil. And that leading to things like disease suppression and insect pest suppression. But there are also interactions between the biology of the soil and the chemistry of the soil, and this leads to things like effective nutrient cycling, or solubilization of the very large phosphorus resources that exist in most soils. Other things like nitrogen fixation, and also carbon storage and organic matter accumulation in soil. There are also biophysical factors, so things like aggregate stability, how well the soil can clump together, and how porous it is and how well water can infiltrate. The biology of the soil also plays a really big role there. The point here is that biological health is really at the core of soil health, and the microbiome is at the core of soil biology. So it's fair to say that conventional agriculture largely overlooks soil biology. A farmer once told me that some time ago, soil was really just seen as something to hold up the plant, but it really is a lot more than that. It may seem obvious, to say, but soil is not a plant. Soil is soil, and what that means is that soil requires more than just NPK. I think a lot of growers think in terms of NPK and maybe trace elements as those critical things they need to provide the plant so that it can grow and thrive. But this is because plants are photosynthetic. Soil is mostly not photosynthetic. There are photosynthetic organisms there, but it's not photosynthetic. And what that means is that a balanced diet for soil needs to include organic carbon inputs, and these can be achieved through a number of different ways. So compost is a good example that I'm sure a lot of avocado growers would be aware of. But there are other kinds of organic amendments as well, but also things like inter-row planting, clever inter-row planting of diverse plants can feed carbon into the soil, and also feed things like nitrogen into the soil as well. The point here is that a productive soil microbiome really requires inputs of organic carbon. This is a critical thing. If the soil microbiome isn't provided this kind of source of organic carbon, then it's in a state of starvation. So the microbiome isn't static. It can be shaped in different ways. And there are a number of things that land managers can do to shape those microbiomes. They can be broadly broken into one of two approaches, either direct shaping of the microbiome, which you could call augmentation, or the indirect shaping of the microbiome, which you would probably call stimulation. What that comes down to is, so direct or augmentation means inoculants. This means pouring live microbes into the soil. The indirect methods are using things you could call biostimulants, or management practices. The way to think of this is that pouring live bacteria onto the soil is like pouring workers into the soil. You're pouring live organisms that are going to hopefully do a job for you into the soil. Providing biostimulants, and biostimulants can be thought of as I mentioned before, compost or other organic amendments. They're providing fuel for those workers, for the microbes there, providing them with food. And management practices have a big role in providing housing, so providing the right structure in the soil for those microbes to thrive. So really what this sort of direct approach means, it's about introducing foreign microbes into the soil, whereas the indirect approach is about encouraging the growth of indigenous microbes. And I think this indirect approach is far more fruitful. There are very few effective inoculants outside of the world of legumes. There really aren't very many, if any, genuinely effective inoculants. So which of these interventions are most likely to be effective? Well, I already named a few, but practically for growers, there's a lot in management that can be fixed. That conventional agriculture leads to a number of different things, like soil compaction, the use of fungicides and fumigants, monoculturing, excessive levels of nitrogen and phosphorus fertilization, which can lead to lowering of soil pH. And things like cultivation or tillage. So talking more directly then about soil and soil microbiology. Soil microbiology really is a tricky thing. So soil is arguably the most complex ecosystem on Earth. Microbes can't be reliably classified visually. They look kind of just like tiny little dots underneath a microscope. So you can't really classify them by looking down a microscope. And most soil microbes can't be grown in culture. About 99% of soil microbes have never been grown in culture. So if this little dot down in the bottom right-hand corner of the screen represents what we know about soil microbes, in terms of those that we've cultured, this big dot represents that which is unknown. And this is where the methods that I use in my lab most often, which is DNA analysis, have really come into their own. Because you can analyze the DNA of creatures without ever growing them in the lab. So the analysis of DNA has really illuminated the immense importance of the soil microbiome. How that works is, if you were to take an example here, we've got four microbes, and we were to sequence a little part of their DNA. So if you don't know, DNA, when you you can read the sequence of DNA, and it's read out and translated into 4 different letters, so A, G, C, and T. In the example here, you can see the 2 microbes in the middle have an identical sequence of A, T, C, G, A, T. But the microbes on the top and the bottom have slightly different sequences. What that means is that based on that information, we can say that we've got 3 different species, for want of a better term, 3 different species of microbes in this mix. So the point here is that DNA analysis can unambiguously identify microbes, and they can be thought of as a little fingerprint for the microbe. And the majority of the examples I'm going to talk about from the literature and from my own research all use this technique. So they use DNA sequence analysis. When it comes to analyzing the plant and the soil microbiome, there's a number of different environments that we can define. So we can analyze just the soil around the plant, or the bulk soil. So that would be the soil microbiome. But we can also analyze the soil that clings to the roots of the plant, and that would be called the rhizosphere microbiome. So rhizosphere just means that area very close to the roots of a plant. But we can also wash all of that soil off and mash up the roots of the plant and analyze the microbes that live inside the roots, and that would be called the root endosphere. Then we can analyze the surface of the leaves, which is called the phyllosphere, and we can analyze the internal parts of the leaf, and that would be called the leaf endosphere. So we can collect all of those different samples, and then we can extract DNA from those samples and then analyze the microbes that are present there. So this next example doesn't involve DNA sequencing, but it's pretty important, I think. So fairly recently, in the last 5 years or so, our understanding of soil organic matter and how it forms has pretty radically changed, or has been challenged at least. The initial idea was that soil organic matter comes from plant residues. So a plant dies, or its root system or its root exudates that it makes get broken down bit by bit, and those bits that can't be broken down, those bits that are recalcitrant, they form the soil organic matter. But some recent new techniques that have changed this in a way is this combines a technique called electron microscopy. So it's looking at very, very fine details within the soil and being able to chemically analyze the soil as well. What they've found is these little red particles are pieces of soil organic matter. And by analyzing the chemical signature of them, what researchers have found is that the vast majority of soil organic matter is microbial in origin. It's not actually plant origin. It may have come from a plant originally, but it's been turned into microbial biomass before it has become soil organic matter. So what that means is that now the soil microbiome is really at the center of soil organic matter and soil organic matter accumulation. So residues come in from plants, from either dying plants on the surface or from roots underground. That organic matter is cycled by and consumed by the soil microbiome and turned into microbial biomass. Some of those nutrients that the microbiome processes get given back and transferred back to plants. Some of it becomes soil organic matter, and some of it gets respired, so turned into carbon dioxide in exactly the same way that we produce carbon dioxide when we breathe. The way that we can influence that is by providing the soil with the right kinds of organic amendments, be that compost or other kinds of biostimulants. We can affect the efficiency of that system. So what the soil microbiome can do is respire, if we encourage the right consortia of microbes to exist in the soil, less of that organic matter that goes into the soil will be burned off as carbon dioxide, and more of it will be turned into soil organic matter. And over time, that can build the pool of soil organic matter. So really the key message there is that managing the soil microbiome is the key to building soil fertility. So now I'd like to give a couple of examples from the literature that I think are just very interesting. And this is an example of how soil microbiomes can be engineered to specifically influence plant physiology. So what these authors did, this is not work that I did, is they took a bunch of plants and they grew them up and selected from them the 4 earliest flowering plants. They used that soil from those plants to inoculate a next generation of plants. And then they repeated that process, picked the 4 earliest flowering, used that soil to inoculate the next generation. And they did that 10 times. They also did the same thing then with the 4 latest flowering plants. So after 10 cycles, after 10 generations of doing this, what they could do is alter the flowering time of the plant by 10 days. So these are very fast growing plants. This is it's a plant called Arabidopsis. It's the lab rat of the plant world. That's what is used by researchers in labs. That's very fast growing, but that's a very significant change in the flowering time that they were able to achieve in a very short time. So what this shows is that the soil microbiome can really significantly alter the flowering time of a plant. So I'll show you a bit of now of the sort of the data that this was all done by DNA sequencing. So the graph you can see on the left here is a way of summarizing the diversity of all of the microbes present. So all of the dots represent an individual plant, and those dots that cluster close together have similar microbiomes, and the dots that are farther apart have more different microbiomes. So what this shows is that the early flowering, the soil of the early flowering microbiome has a distinct signature, as does the late flowering microbiome. They then went and analysed this further, and this graph might look a bit confusing, but I'll try to make it a bit more simple for you. So I've drawn a box over, these are all of the early flowering plants and the late flowering plants. And as you go down the graph, each one of those little lines are different bacteria. What this shows is that the early flowering plants were enriched in the bacteria that are coloured blue. So the dark blue colour means there were lots of those particular microbes. The pink or the red colour means there weren't many of those microbes. So what's happened here is that the early flowering plants are enriched in a certain set of microbes, but depleted in another set of microbes. And the late flowering plants had the opposite. So this showed really, really clearly that these microbes were having a clear impact on the flowering time of the plant. And this could be something that could be implemented, probably not in an orchard. You know, transplanting soils in an orchard is not particularly practical, but in a nursery setting, this could be something that could come into play and could maybe lead to having varieties of avocados being produced that could have different flowering times, which could be useful from an economic perspective. In a similar way, disease suppressive soils are also a product of the soil microbiome. So these authors looked at numerous data sets to see if disease suppressive soils had a predictable microbial signature. What they found was that this was indeed the case. So they looked at, I think, 24 or 46 different soils, different studies that looked at a number of different soils, and found that these particular microbes, you don't need to worry about the names. Some of you may have heard of streptomyces. It's a very common microbe that is known to suppress plant disease, but also some other microbes that are less commonly known, but recent literature is showing that they play a big role, it seems, in suppressing plant diseases. The point here is what they found is that they found a particular signature of disease suppressive soils and showed that these suppressive soils are really a product of the microbiome. Then this is another study about disease suppressive soils, and what these authors showed is that, similar to the flowering time study, the authors showed here that disease suppressive characteristics could be transplanted. Again, that's not particularly useful for people who are managing an orchard. It might be more useful for people who are managing nurseries. But just because these things can be transplanted doesn't mean they can't be cultivated. Once we understand the right microbes that we need to encourage, that's one big piece of the puzzle and gives us a direction in which to try to manage our soil microbiomes to cultivate particular plant outcomes that we want, be it suppression of disease or as in this example, perhaps suppressing above ground pests. So it might seem a bit bizarre or counterintuitive, but soil microbiomes can actually suppress the action of insects that are active above ground, that are active in the canopy of the tree. So this can occur by a couple of different ways. The one way is recruitment of endophytic microbes from the soil. And Metarhizium is an example there. This is a fungus that parasitizes and kills insects. But another way is that the microbes in the soil can stimulate the plant defensive responses. So the jasmonic acid pathway is kind of the technical name for one of those pathways. But microbes in the soil can stimulate these pathways and in that way suppress these above ground insect pests. So the take home message here is that the microbiome can really mediate suppression of insect pests. So I thought now I'd give a couple of examples from the research we've been doing at Metagen. So this is the information I'm going to present here is DNA sequencing data, but it comes from analysing the root endosphere. So we collected the roots of the plants, washed them off, and then mushed up the roots, the cleaned roots, and analysed the microbes that were present inside the roots. And this was work that was done as part of our product development research. So we had one study. So the way that this works is that we had treated versus untreated plots. So we had the treated plots were treated with our research products, and then we had control sites that weren't treated at all. So in avocado, what we saw was a 65-fold reduction in Phytophthora. It's worth noting here that this particular sample was a pretty sick plant. So while we think that this kind of reduction in Phytophthora is impressive, it was coming from a fairly sick plant. What we also noticed was significant increases in a couple of different fungi that are beneficial. So the names there, you don't really need to worry about them, but Rhizophagus is what we would call a mycorrhizal fungus. So these are fungi that make associations with the roots of plants and help them to acquire nutrients, but can also protect them from plant diseases. And the last one there is a fungi called Cladosporium, which frequently adopts a fungal parasite life strategy. So it parasitizes and eats other fungi. In sugarcane, what we saw were increases in a number of different bacteria. So the bacteria at the top here is called Mesorhizobium. It's commonly associated with chickpeas. It's the rhizobium that forms root nodules and fixes nitrogen in chickpeas. But these species are also found in soil generally and in a whole range of other places. What we think is happening here is that the Mesorhizobium is living inside the roots and maybe fixing nitrogen internally for the sugarcane. So acting in some way kind of like a legume, not in root nodules, but still providing nitrogen for that plant. The next two microbes there are Pseudomonas and Sphingomonas. These microbes are involved in producing enzymes like phosphatase is an example that can solubilize phosphorus from the soil and provide that as a nutrient to the plant, but are also very active in disease suppression as well. And the last example here is from green beans. The microbe at the top there, Variovorax, is a really powerful suppressor of plant disease. And the microbe at the bottom there, I won't bother saying the name, but this is a microbe that can convert nitrogen fertilizer into nitrous oxide and nitrogen gas. So seeing a reduction there is you would want to see a reduction in these kinds of microbes because they're the sorts of microbes that are removing the nitrogen inputs from the soil, making greenhouse gases in the as a byproduct of that. So the point here is that this was just from analysing a small set of soils, and we've analysed many soils since. So we've got a number of other research projects ongoing at the moment, but we're not presenting the results of those there. But the more soils that we analyse, the stronger the databases and data sets that we generate, and the better the predictions that we can make. Also, the more metadata we collect about these samples, so following up and seeing if there's reduced disease impacts as a result of these kinds of treatments is key. The point here as well is that we wouldn't be able to do this without DNA analysis. So this led us, the focus of our research at Metagen for the last couple of years has been the idea of broadening this DNA analysis a bit further to come up with a soil health report or a soil health report card. So to do that, we've built a lab in our factory in Gatton in Queensland. This is a picture of the lab with my colleague Ken Fitzgerald, who's the Director of Production at Metagen, and one of our collaborators, Graham Sterling, who's an eminent nematologist, so studies nematodes. So the way that we present these, or the way that we present these soil health reports is we do give a single value, so an overall value. But the point here is that soil health really can't be reduced to a single score. These are some of the limitations with other tests that are touted as soil health reports, like Solvita or soil respiration. This gives you a single value, but you can't really. Soil health is so multifaceted that you can't really give it a single value. So what DNA analysis allows us to do is to dig down deeper and to pull out a number of different useful bits of information from the data that we gather. So this is the more detailed version of our soil health report that it has a number of different things. So I'll take you through them a little bit one by one. So one is a stability index. This gives you an idea of whether or not the soil is in a state of flux. So has it undergone a recent disturbance or perhaps a recent application of fertilizer, something like that. Then because we capture all of the bacteria and fungi that are present in the sample, but also some of the higher organisms as well, so things like nematodes and protozoa, we can do what we'd call a soil food web analysis. So we can analyze the predators, higher and lower predators that are present in the soil, as well as the consumers or the primary producers, so the bacteria and the fungi. So a healthy soil ecosystem will have pretty good abundance of all of these different groups. We also analyze chemically, we analyze phosphatase activity. So this is the phosphorus. Phosphatase is what's known as a phosphorus solubilizing enzyme. So these are proteins that are made by microbes that get released into the soil and can solubilize the phosphorus that's become immobilized in the soil and make it plant available. There's something we call the bacteria efficiency index, and this refers to the potential for soil carbon accumulation. So the more efficient the microbes that are present in the soil, the more likely they are to be creating biomass and generating soil carbon reserves, as opposed to burning off and releasing a lot of that organic matter as carbon dioxide. We can also specifically measure mycorrhizal fungi, so the diversity of mycorrhizal fungi. They're those fungi that make beneficial associations with the roots of plants, which I understand can be, we think are particularly important for avocado plants because they don't have root hairs. We then also have a couple of biologically relevant chemical and physical properties. So we measure aggregate stability. So this is when you take an aggregate of soil, how quickly does it break down and fall apart? And we measure something called active carbons. This is what we'd call either active carbon or labile carbon. It's the carbon in the soil that serves as a ready source of food for the soil microbiome. Then we also have a couple of indices that measure the potential for disease suppression in the soil. So we have a suppressive bacteria index, and this is measuring the diversity of many of the microbes that were mentioned in that previous slide about disease suppression in soils, and a bunch of others as well. So we look at the diversity of all of those microbes that we know can suppress suppress plant disease, and then develop a score from that. But also another enzyme which is made by the microbes and released into the soil, which is a cellulose degrading enzyme, which is in the literature been linked heavily to, or linked strongly to, disease suppression. So I should point out that this actual example soil report we've got here is from an avocado plantation. It was a single sample sent to us where the grower was interested. It was a diseased sample. So we're able to show here that perhaps the suppressive bacteria index was a bit low, but we were also able to detect Phytophthora in the sample. So we could point to the grower that yes, Phytophthora was a problem in this soil. So I'll finish up by saying maybe a bit about what the future might look like. And agriculture, it faces a number of challenges, and the biggest challenge really is to be able to grow a lot more, but do it with less, and preferably to do it sustainably. And ultimately, the solutions for this have to come from technological innovation. But fortunately, there are a number of disruptive technologies that are on the horizon. So precision agriculture is one I'm sure most of you will have heard of. This idea of big data, as well as automation and robotics, and microbial ecology, the microbiome. So precision agriculture means we can collect data better and we can use resources more efficiently. Big data, so being able to analyze really big data sets, helps dealing with the complexity of agriculture. It's an extremely complex thing. Dealing with living ecosystems is incredibly complex, but it also helps us to find hidden trends. So many of the examples I've given today are examples of finding hidden trends in very complex data. So DNA data that we get hold of is extremely complex, and only with big data analytical techniques can we find these associations between the soil microbiome and particular plant outcomes. So automation and robotics, well, that's you know, it's going to lead to perhaps the miniaturization of agricultural equipment, but also importantly, reduced labour. And microbial ecology, really, I think, you know, I'm not biased at all, but offers the most exciting revolution for agriculture. But what it's about providing biological and ecological solutions. And all of the problems in agriculture, at least in terms of agricultural production, they're all biological problems or ecological problems. And ideally, biological and ecological problems should have biological solutions. So I think farming in the future is likely to be less labour intensive, and this is going to give growers more time to spend monitoring information and formulating management responses. And I think that the management of the soil microbiome to improve soil health is going to play a huge role in all of this. It's not the only thing, but it's going to play a huge role. And I think I'll finish on that note and say thank you. But what's on this slide here are a few books. If any of you are interested in reading a little bit more about the importance of microbes, that first one, How Microbes Can Help Feed the Earth, it's freely available online. If you Google that, you'll find it. And that's a booklet made by, I think it's the American Microbiological Society, and it's all about microbes in agriculture. The other two books there are more broad. There's a lot about microbes and humans and other things, but they make fascinating reads. And on that note, I'll say thanks very much.
Excellent. Thank you, Neil, for a fascinating talk. Certainly given us all a lot of food for thought. Yeah. Neil, could you just first off turn your video on so we can see you for the question time? And we've got a lot of questions. Just to remind people that this presentation has been recorded and it will be available on the best practice resource. I know there's a few questions there asking about maybe repeating some things that you covered. Obviously, we can't do that right now, but you can watch the recording at leisure once we've got it up there. And also, we will be providing Neil's contact details in the minutes as well. Okay. Is Neil goes video on? Yeah. Okay, I'll go to the start of the questions. We've got about 10 minutes. Right. Good afternoon, Neil. Neil, presumably I'd like you to explain the interaction of the soil/plant microbiome and beneficial endophytes, both bacterial and fungal. And I will point out that question was posted quite early in the piece. You might have explained some of that already. Over to you, Neil.
Yeah. So there's some evidence that plants can actually carry endophytes in their seeds, and they can pass them on from kind of mother plant to daughter plant. But the main source of plants will recruit their endophytes from the soil. So generally, the soil serves as a reservoir for the recruitment of endophytes. So I guess what you tend to find is that the soil is incredibly diverse with lots of microbes. The rhizosphere, so that area just around the root, gets a little bit less diverse. And then the endosphere is a lot less diverse. But recruitment generally goes in that direction. So from the soil to the rhizosphere into the plant. So if you don't have the right kind of microbes in the soil, generally you won't have the right kinds of endophytes in your plants.
Thank you, Neil. The next question, I'd like to know which cover crops / green manure crops for orchards would be recommended in regard to soil health and to avoid having to spray against weeds and to help nitrogen in the soil.
Right, that's probably a bit out of my my area of expertise. I don't know that I can provide a really good answer to that one, although, so including things like legumes in the mix are a very good idea. Also including things that have deep rooting and shallow rooting is a very good idea. So the idea of having good inter-row cropping is again, it's about diversity. So there are certain plants, I think buckwheat is an example, that is particularly good at producing phosphatases. So it's good at releasing phosphorus for the soil and for the plants that you're trying to grow. Unfortunately, I'm not sure. I can't name something that might be useful for suppressing weeds. That would probably be more a question for a botanist or a yeah. I think there was something recently I was sent. I haven't been able to access it, but I think the man's name is Richard Llewellyn, who had provided a video. I can see about getting hold of it for the person who asked the question about this idea of inter-row cropping and the sorts of plants to introduce.
Thank you, Neil. I think you've given some good direction there. Next question. Some of our soils can be very low in mycorrhiza. There are some good commercial inoculants now, like Endoprime. These have been positive responses. I think there have been positive responses to these AMF inoculants in many crops, including avocados. Do you believe AMF inoculants are effective?
Not that I've seen, if I'm honest. I've heard positive reports from growers. The problem with AMF, the problem with mycorrhizal fungi, is they are very difficult to grow. So they're very difficult to make good quality inoculants. So you can't grow up the fungus on its own in a big culture vessel. You have to grow plants pretty much to grow the mycorrhizal fungi. This makes them very difficult to produce. So I'm a bit skeptical of AMF inoculants. But if you see a particularly good response with one, I'm not going to dispute that. It's just that they are difficult to make, and so there are a lot of poor quality inoculants out there. I'm not familiar with the particular brand that you mentioned there. You know, the only way to know, really, you know, if your mycorrhizal inoculants have been successful, there are ways to analyze the roots of the plant microscopically, or you could always do DNA analysis to see if what you're doing is actually increasing the abundance or the diversity of mycorrhizae in your soil.
Thanks, Neil. Next question. Could you explain the difference in endophytic organisms, rhizospheric ones and soil dwellers? What are the benefits of each one?
Right. So soil-dwelling organisms are going to have a pretty limited impact on the plant. Their impacts are going to be on the soil. Rhizosphere microorganisms are going to, by their nature, by their physical proximity to the roots, are going to interact with the plant more. So they're likely to play more of a role in things like disease suppression or protection of the plant from disease, but also providing the plant with nutrients, but also things like stimulating these kinds of responses to, so say, to above ground pests or something like flowering time or other physiological things. Endophytes, they have a truly intimate relationship with the plant, and it's arguable that some people think that endophytes are perhaps the most important. I think that that all are important in different ways. So, an example of an important endophyte would be those fungi that can colonize the plant and therefore protect the plant against above ground pests or above ground diseases. But all of them are important.
Thanks, Neil. Next question. Can you comment on the effect that glyphosate and herbicides in general may have on the soil microbiome?
There's fairly limited information about the effect of glyphosate on the soil microbiome. So I know that glyphosate can be broken down by microbes. It's not a particularly sort of what we'd say a particularly recalcitrant compound. So it's actually broken down by microbes in the soil. So there are microbes that can use it as an energy source. I'm not really familiar with a lot of the literature. There certainly has been studies that have looked at the effects, but to my knowledge, the glyphosate, I'm not sure about other. A lot of the focus is on glyphosate, has a pretty limited impact on the soil microbiome. That's what I can say there. I wouldn't be too concerned about its impact on the soil microbiome.
Thanks for that. We're running out of time. We'll have a couple of quick questions, and then we'll see if Neil can answer the others via email for the minutes. Can foliar applied fungicide sprays for anthracnose and Phytophthora a negative effect on the beneficial fungi in the soil?
Yes, so I had a slide actually from there. So I did a little bit of reading on so phosphonates, which I know are really commonly used. They seem to be pretty good. So phosphonates are generally environmentally benign, and they're broken down really rapidly by soil microorganisms. So their effect is likely to be pretty limited on the soil microbiome. And there's some evidence that that phosphonates are actually, they're more effective against oomycetes, so more effective against things like Phytophthora than they are against other microbes. But phosphonates are also used against some other fungal and bacterial pathogens. So the thing about fungicides, about all fungicides, is that they're not surgical. They don't have surgical precision. They're generally pretty broad. You know, they have a pretty bad impact across the board on all fungi or all bacteria. But phosphonates seem to be probably the better of the lot. Copper, certainly, at sufficiently high levels, copper has a strong negative impact on soil biology. And that effect is usually seen in terms of reducing the diversity of the soil microbiome.
Thanks, Neil. I'm going to have to cut the questions off now. We've got a lot more questions, but we just don't have time to answer them right now. But as I said before, Neil will answer these, and we'll provide those questions and answers in the minutes.
Happily. Yeah.
Thank you very much indeed, Neil. We really appreciate your very interesting talk and answering those questions so well.
No worries. It's my pleasure.
Thank you. All right. So, just bear with us, everyone. Someone else sharing. Who else is sharing their screen, Ron?
We're sharing now.
Okay. Can you hear me? Yep. Okay, so thank you again, Neil. Now we're going to call upon Andrew Robson and Craig Shephard, and they are from the University of New England, and they are part of the Applied Agricultural Remote Sensing Centre. And they're going to give us an update on a very interesting project on precision agriculture that they're undertaking on behalf of the industry. So I'll hand over to them.
Hey, Simon, can you hear me all right?
I can, yep.
Okay, I'm just sharing the screen now. And share. Is that all good?
Yep.
Cool, excellent. I assume you don't need to see me, so I'll leave the video off unless you really want to. Okay, so yeah, my name is Andrew Robson. We've been working on validating predominantly remote sensing over avocado for the last, nearly 7 years now, to try and work out how robust certain imagery formats, so satellite, drone, ground-based, robotics, a range of platforms with a range of really good expertise across Australia from different DPIs and universities to see what works. A lot of you in recent times would have probably been blanket called by a lot of people selling imagery. We can at least say that we've done a lot of groundwork to sort of identify what works and what doesn't. Anyway, currently through Hort Innovation and Avocado Australia funded project, we're looking at a couple of outcomes to further produce those results and bring them to you. So one of the objectives of this project is to update the national map of avocados along with other industries, and I'll leave that to Craig Shephard and Joel to talk about after me, but that's one of the objectives. The next one is to further validate the relationship between remote sensing or measurements of canopy reflectance and yield parameters, so fruit weights, tonnes per hectare, fruit number, but also better recording tree health as well. So we're trying to understand the influence of location, season, variety on that relationship. We also wanted to bring that information better to growers. So we've worked with a group called Circulate, app developing group out of Sydney, to bring you a format which brings basically the self-calibration work to do yield forecasting and yield mapping in the field. And the idea is to include a range of different imagery sources, including some freely available ones. So again, it's based on a lot of field work to try and get to this point. It's not just another app, hopefully. And the fourth one is, it's obviously to go out and do some calibration work is sometimes considered a pain. And so we're trying to work out ways to better forecast at the block, farm and regional level without the need for actually counting fruit. And so we're looking at integrating the national map, as I've mentioned that Craig and Joel will talk about in a minute, but also our understanding of remote sensing and historic growth profiles. So it's basically how your orchard is responding over the last 5 or 10 years in terms of crop reflectance, how that relates to the yield that was achieved by those blocks in those corresponding years, and also the inclusion of weather information. And I'll talk very briefly on an example in macadamia we're achieving that at the moment. So as I mentioned, this is a continuation of a lot of time and work that went into a lot of field trials that went in over 3 years. And that itself was a follow-on some early work with QDAF on avocados in the Queensland growing areas. So we did this work, and as I said, we trialled a range of different on-ground robotics and drones and planes and bloody carry pigeons. And yeah, we come up with a methodology that we believe works, or has shown to work really well, or better than current practice in the areas that we've evaluated. And so we reckon it's a pretty strong methodology that we're looking at bringing out more to US growers. And does it work? So we got some examples, and some of you might have seen these from the field days previously. But here's 2016 with a number of blocks, 5 different blocks. The dark bands on the dark colours are the actual yield achieved by the blocks in tonnes per hectare. The middle one is how we predicted from imagery, and the last one is comparison to the current eyeball estimate. And it gives you a comparison of the imagery approach works quite well, 2016. 2017 was a real eye-opener. Like some of the blocks that we were working with, the eyeball or commercial forecasting method was shown to be, in some cases, quite heavily over-predicted, or in some cases, quite heavily under-predicted. Obviously, if you're making decisions about labour, storage, packing, market, selling and things like that, these sort of differences could be quite large. And I'm going to show you the little crop count flick or little movie in a minute, which will explain the actual, put some dollars on how much that sort of discrepancy can cost. But in comparison, our forecasting using the methodology that we've developed is quite good. So we've got lots of numbers to back up what we've done. So as well as doing a forecast of the average yield for those blocks, we also use the relationships developed from the canopy reflectance and canopy size to yield achieved, and we turn that into yield maps. So in terms of kilograms per tree, and you can see there's a range of kilograms per tree from the low producing ones to the high producing ones, and then also reproducing it the following year and seeing how much that yield variation has changed over time. And also with avocados, the influence of irregular bearing is quite strong, and that's why we've sort of led to that need to do that calibration work. And so the benefits, obviously, of identifying yield variability, such as this, means that you can do targeted agronomics to work out the drivers of low production areas. It's been shown to be a really good indicator of phytophthora, of where large phytophthora areas are occurring in predominantly wet years. The positioning of soil moisture sensors and things like that, and leaf tissue testing. We've also found strong relationships to fruit size as well. And so these maps are obviously the same block from the same years, but the maps are now produced in the grams, or the average fruit size per tree. Again, this is pretty important if you have to meet a market requirement of a certain fruit size. So instead of selectively or harvesting every row and selectively picking certain trees, or alternatively picking a whole block and then sorting them in a pack house, if you're pushed for time, it's a good option to target your harvesting in these areas that may better meet the requirements of the supermarkets or whatever you're selling to. So currently, in the current project, we've developed this crop count app, which is trying to, as I mentioned, bring this methodology to growers. So we've developed the prototype, and I'll show you, as I mentioned, a little short flick in a minute. And we've run it with 3 growers initially, and that was the scope of the project. So we were limited to Queensland in the first instance to see how it goes. And so we were very fortunate to have the engagement of Blue Sky Produce, Simpson Farms and Auschili to work with us last season and this season as well. So now I'm going to show you the little the little clip of crop count, which better explains it. And Hort Innovation got us to go through the GATE program through New South Wales DPI as part of developing this sort of this fast streaming of R&D to commercialisation. So I'll just play this now, and hopefully it comes across all right. I've been told there might be a little bit of a lag, so apologies.
Look at this magnificent avocado farm. I want you to imagine it's your avocado farm. You need to know how many fruit you're going to pull off those trees for harvest this season. It's not an easy job. Look at all those trees. Would you count all of them or just some? Yield forecasting, which is knowing how many fruit you're going to get each season, is incredibly time consuming. One tree can take 5 minutes to count because it's so large and there can be up to 400 fruit on that tree. This means that you will need 3 people to count that same tree to ensure accuracy. And on a medium-sized farm, which is about 10,000 trees, the current practices count between 70 to 1,000 trees, and it has to happen twice a season. So all up, this is about 500 hours of effort just to get a yield forecast. And even after all that time, it can still be incredibly inaccurate. This 2017 trial shows on the left what the farmer thought they were going to get, and on the right what was actually harvested from the trees. In all of the blocks, they're almost over 120% out. The impact of this is huge. Just taking one of these blocks, which is a 30-hectare block, you can see the farmer thought they would get 9 tonnes per hectare, but they actually only got 4 tonnes per hectare. This equates to over $1.5 million when their estimate is out. So the flow-on effect of this to their labour costs, the price they can get at the farm gate, their ability to fulfil existing contracts and their packing costs is huge. And this is just 30 hectares. We know that in Australia, there's over 13,000 hectares of avocado orchards. Introducing crop count. Crop count combines satellite imagery with on-the-ground sampling to produce yield forecasts. And the big difference we make is in the time saving. So previously we were talking about counting between 70 to 1,000 trees. We've reduced this to just 18 trees. That means the time saving is from 500 hours right down to 9 hours. What previously took almost 2.5 months can be done in almost just 1 day. And what about accuracy? As shown in that 2017 trial, crop count is 93% accurate. So this is more than just an idea in our heads. With the help of Hort Innovation, we've created a prototype mobile app and put it into the hands of growers. And what those trials have shown us is that the app is super easy to use. It can be picked up by even a novice with no training and easily used in the field. Team Crop Count is led by Professor Andrew Robson from the University of New England. He has over 25 years of experience and is the leading researcher in this field. Andrew Robson identified a need to partner with a team with expertise in digital product development. And so here at Circulate, me, Janine and Alana brought that expertise. We've tested for 5 years across different farms to ensure that the scientific model is accurate. And over the past year, with the support of Hort Innovation and Avocado Australia, we've created a prototype app to put this scientific model into the hands of growers. Our next phase is to build components of the back end to ensure the scalability of crop count. And we'd like to do more trials to look at how we can extend this into other industries. We're looking for partners to ensure the crop count is ready to be fully commercialised and hit the market. There's also an exciting opportunity to see crop count extended into other industries such as citrus, mango and macadamias. We'd like to thank Hort Innovation for their support in getting crop count to the prototype stage of that, and we're really excited about the next phase. Crop count provides accurate, efficient and consistent yield forecasts.
Okay, hopefully you can still hear me all right. That all worked. So that gives you an idea, the glossy version of how it works. So the 2020 outcomes was the app testing on 3 farms that was undertaken at Final Fruit Set. to do the calibration, and also at pre-harvest, or both in some cases. We modified the sampling regime, so we initially had the trees spread across the orchards, and we ended up grouping them in 3 clusters, which reduced the sampling time down to 40 minutes per block. The accuracies from pre-harvest were really good across all blocks, so that's the ground truthing work done just prior to harvest, when obviously the fruit are largest and biggest to see. But at final fruit set, when the fruit are a lot smaller, we achieved good results in the north up in Jimbula with a lot smaller trees, but it did struggle in big trees. And there's the example of estimating the number of fruit per trees done at fruit set by the grower, Matt Feely up north. And as I said, he put a lot of time into doing those counts on those trees, and then it reflected in the accuracies. So as I mentioned before, sampling can be seen as a bit of a pain, and so we're looking at investigating the potential predicting the yield at the block farm regional level without sampling. So this is the integration of historic production information at the block level, obviously the farmer information, but also within season growth profiles from remote sensing and climate. We do have a PhD student working on this, and currently he's done a review of all these attempts across the world in different tree crops, including avocado, and within avocado it's quite non-existent. So to achieve this, we need to access historic block information from a range of growing areas to test in the model. So we need you guys to help us help you approach. Remembering that better yield forecasting methodologies were an outcome requested from the industry initially, which drove this research. So currently in macadamias, it's worth showing you that we do have a lot of information starting coming from growers across different regions. We're running a similar approach to compare those same, the national map, historic growth profiles, historic productivity, and climate information. And in this case, macadamias do have a climate model existing that we've used. The accuracies thus far are looking really good. So we're providing forecasts at the block level 5 months before harvest without sampling. And here we have actual yield at the block level for 3 different growing regions versus the yield achieved. You can see it's over predicting the really high yielding blocks, but generally the block level forecasting is really encouraging, as this is the first year. And again, I said that's over 3 regions. We're also delivering those block level forecasts through freely available, again, web apps to show their forecast. And again, this is 5 months before harvest. So we're really encouraged for this, and we want to do that for avocados as well. But we really need engagement from you guys as well. Okay, that's all from me. I'm going to throw it over to Craig and Joel now to talk about the mapping side. So I'm going to stop sharing and let them take it on.
Good afternoon all. Hopefully you can hear me now and see my screen.
All good.
All right, I'll kick off. So it's Craig Shephard speaking, and I'm joined by my colleague Joel McKenzie in the corner. If you have any questions as we undertake this presentation, get them in the chat and Joel will respond accordingly. But otherwise, hoping to introduce you now to the phase 1 and now known as phase 2 mapping update of the Australian tree crop map. I'll run through some of the methodology that goes into building the map and principally focus on the applications that you as growers and your industry can now access. So the phase 1 map was first published in 2017, and it sought to build a baseline mapping product of all commercial avocado, macadamia and mango orchards across Australia. When I say commercial, I'm referring to the scale of the mapping, and that then was a minimum mapping unit of 2 hectares. Let's bear in mind that the information presented in the Australian tree crop map is simply tree crop type, location and extent. There's no personal or commercial information contained within, and blocks are aggregated relative to or regardless of property boundaries because it's simply not required. Moving forward to phase 2, we're now updating that map, principally because of the high growth in horticulture tree crops around Australia, but also to additionally include all citrus, olive and banana plantations. The scale of the map is also changing in that we're engineering in more detail, and this was at the request of stakeholders and industry. The minimum mapping unit is now 1 hectare. The mapping presented here is shown as clusters, clusters of points, features. And we do that because at the scale, you cannot visually see the orchards. As you interact with the map and zoom in, the polygons will reveal themselves such that you can see the tree crops. It's important to understand that the scale still fits a national map of 1 hectare. Okay, let's look at what goes into making the map. So we're lucky to have the collaboration with our land use mapping colleagues in all states and territories around Australia, and that's through that ACLUMP group. So we use existing land use information to inform our mapping update. You need to understand that the currency of that data and the consistency across Australia is really mixed. Some of that information is dated to 2003, hence the need to update the map into the Australian tree crop map. It's also informed greatly by industry collaboration, and that's where we can engage growers, local experts and the like to assist us in building the map. And shown here is the land use survey observations which have been submitted. So Joel and I will interpret that information at our desktops, but the principal component of building the map is the imagery. So we use multiple sources of open source imagery from a variety of platforms and sensors. And here's a sample of 4 different orchards for you. And what I'd like you to understand is that land management practice varies, which makes our imagery interpretation of tree crops very challenging, because all the crops here are avocado. And look how different they are. We also use open source Street View imagery to inform our mapping, and this is available to anyone through the Google Maps application, which greatly assists us at the desktop. The other important component in informing our mapping update is the ground truthing or fieldwork. So this greatly improves the thematic accuracy, and that refers to correctly classifying crops from one another. What it's also done is aided us in mapping the new crops because we can't see them yet in imagery. Here's a great example of that, where we've got an orchard map that's avocado, yet I can assure you the imagery beneath this feature is likely sugarcane. The 2 land use observations shown there are indeed feature avocado surveys, and here's an image that's been submitted with that survey of the new trees in the ground. So really, really important. So we'll cut to the chase, and what are the outputs? Well, it's a spatial data set of location, extent and tree crop type. Nowadays, we deliver that information through a variety of web-enabled GIS maps, apps and dashboards. You do not need a GIS application on your desktop to view this information. It all runs in a browser. These are all available for you to interact with in your own time, and I encourage you to visit that URL and make the most of it. They're all there for your use. I'll skip through some now. Be aware that they each feature the Australian tree crop map, but the context of delivering it is to support individual themes. So one of those themes is industry demographics. Everybody would like to know how much and where, particularly across regions. And so this is the latest update to our Australian tree crop map dashboard. Some of the features within this can be viewed within this little YouTube video, and I encourage you to look at that in your own time following. Interacting with this map, the little widgets on the bottom will individually update relative to the map view extent and summarise the area of orchards within. So it's very powerful. Additionally, I'll just send back to Australia. Pop-ups are enabled so that you can click on a feature, and at this scale it will show states and territories, and again return the metrics for orchards across each state and territory. I'll note that in Janine's video, I think she mentioned 13,000 hectares, so you should also have vote you've crossed the 14,000 threshold. What we're also able to deliver is demographics for change. And these are informing industry, particularly in response to the Paradise Dam issue in Bundaberg. And as we've now updated the map in Queensland, we can present these changes on a map. Note that these only exist for the 3 crops which were mapped in phase 1. So we come into Bundaberg. Any feature outlined in white is a new crop. The mapping greatly informed the debate in Paradise Dam with the extent of new orchards going in the ground. Similarly, we compare to Childers. I'll take you on a little ride around Queensland. And this is showing all avocado, macadamia and mango new crops. But plenty going on. Another application is natural disaster response and recovery. So the mapping greatly informed the recent bushfire crisis across Australia, unprecedented in a sense that it's across states and territories. So this web map again is available from the gallery. And effectively it presents the burn area extent, and that allows users to interact with that layer relative to the spatial location of the tree crops to understand the impacts. And there's a little olive grove, which is particularly vulnerable. The Applied Agriculture Remote Sensing Centre has just launched our latest app, the severe weather one, which makes use of authoritative data from the Bureau of Met, principally in near real time, to deliver severe weather information. The really neat thing that I enjoy about this mapping is the satellite image shown there is captured over Australia in the last 10 minutes, and we'll continue to update. Other layers are available for your use. You'll be familiar with the rain radar, but in particular, we're interested in the severe thunderstorm cells. There's some up here passing through now. So I encourage you to interact with that. Obviously, we're anticipating cyclone season, where this app will really be becoming valuable for industry and your growers. With a little bit more R&D, we hope to build in time-enabled layers such that you can look at events that occurred yesterday and the preceding event days. Some of the challenges we're facing in phase 2, well, it really is the new crops, but here's where you can help. So off our web app gallery, there's one of the tools there is an industry engagement web app. And this is best to inform your use and for you to contribute from your desktop. And you can actually peer review our draft mapping. And here's an example of that in Bundaberg, where features have been added. Well, this was a future one for macadamia. And it brings that information to the attention of Joel and I, which we can then interpret in updating the map. There's a mobile application as well, and that's the land use survey. And here's a selection of those features over Bundaberg. It's really powerful, very easy to use. The land use survey is built for your mobile device. You just need to be connected and you can contribute your land use survey information. We recently were really pleased to see the effort someone has gone to in Mencham up in WA in engineering all these features through the industry engagement web app. So Joel and I will action these in due course when we update that region. So let's look at the mapping progress. Queensland has now just been updated. So the majority of that mapping is dated 2020. So it's a very current picture of the location and extent of avocado orchards across Queensland. South Australia was in progress, and Joel and I were fortunate to get out of Adelaide last week, to avoid the mandatory hotel quarantine. So we hope to get back there and validate the mapping in the field for Renmark in the new year. In the meantime, we're focused on drafting the updated mapping for the Northern Rivers, New South Wales, and there are some avocado orchards to get through. We're already aware of the Combine region where there's particular many new crops going in the ground. Thanks very much for your time. Our email addresses are there and happy to take questions in the chat. But otherwise, thank you, Simon and Brody, for hosting, and I hope to see you on the map soon.
Thank you very much, Craig, and thanks to Joel in the background and of course to Andrew. Some amazing technology becoming available, obviously going to be very useful and powerful for the industry. So thank you very much indeed for that. We've kind of run out of time. Some of the addresses for those maps and things are put up in the chat box, but we'll include all of that information in the minutes. And I just wanted to point out that, again, reiterate that Andrew and his team are looking for growers with historical data and current data to help them with the development of this tool. I think I'm correct in saying that. Right, okay. I don't think there's any specific questions, but if there are, we'll answer them via email. But I just wanted to wind it up by a couple of little items. All right. Okay, so just to wind things up, just to remind you, Noel Ainsworth had the supply chain project. He's done a couple of videos. They're available in the information that's been sent to you. Those areas that are still flowering or about to flower, please keep a lookout for the citrus blossom bug. And if you find some, the project leaders for that new project would love to get some specimens from you. Again, the link to that information of how you send those specimens in is supplied. And probably last but not least, we really need your evaluation, your feedback. There's a little feedback sheet. It'll only take you 2 or 3 minutes. We really appreciate it if you could fill that in. There's a link to that provided. And just to remind you that all this information, so the PowerPoints when we have access to them, the recording of the whole webinar, and the minutes once we've written them, will all be available on the best practice resource as usual. So again, thanks to Neil Wilson, Andrew Robson, Craig Shephard, Joel McKenzie, and all the Avocados Australia staff and DAF staff behind the scenes, and of course acknowledge our funding for this project. So with that, say cheerio and have a happy Christmas.